Hello students, in today's class we are going to learn about photosynthesis. So photosynthesis is basically the process of conversion of light energy to chemical energy in the form of ATP and reducing equivalents. So the organisms that carry out photosynthesis are known as phototrophs. They can be further categorized into two types, photoautotrophs and photoheterotrophs. Photoautotrophs are those organisms that can fix their own carbon dioxide to sugars, while photoheterotrophs are those that make use of ready-made fixed form of sugars. So photosynthetic pigments are important components of the photosynthetic mechanism, and chlorophyll A molecule, it is an important pigment in case of higher plants, most algae and cyanobacteria. It is green in color and it absorbs red light at 680 nanometer and blue light at 430 nanometer and emits green light. In addition to the chlorophyll A molecule in case of plants, there are many other chlorophyll molecules as well, such as the bacteriochlorophylls. Chlorophyll B molecule absorbs at a different wavelength at about 660 nanometer rather than 680 nanometer. So chlorophyll B molecule is present also in plants along with chlorophyll A molecule. While the bacteriochlorophyll molecule will absorb at a different wavelength around 800 to 925 nanometer. So basically organisms have several different types of chlorophyll molecules, and each of the molecule is absorbing light energy at different wavelengths. So this allows the living system to make better use of the energy of the available electromagnetic spectrum. So in case of bacteria, bacteriochlorophyll is important or the central photosynthetic pigment, while there are other chlorophyll molecules such as the antinechlorophyll molecules that function majorly for harvesting light and transferring the light energy to the reaction center chlorophyll molecule. Then other pigments known as accessory pigments such as carotenoids are also involved. So these absorb the blue spectrum of light and they are responsible for the different brilliant colors of bacteria such as purple, pink, green, yellow, and brown. These carotenoids also have a photoprotective role because of their antioxidant property, by which any toxic oxygen molecule that is generated during the process is being used up by the carotenoids. Then we have other pigments such as phycobilins and phycobilizones present in case of cyanobacteria and red algae. These consist of the red pigment phycoerythrin that absorbs at 550 nanometer, phycocyanin that absorbs at around 620 nanometer, and allophycocyanin that absorbs at around 650 nanometer. So these phycobilizomes, they are present kind of adjacent to or attached to the photosynthetic membrane in such a way that these make direct contact with the chlorophyll reaction center so that the energy can be immediately transferred to the reaction center chlorophyll molecule. Now coming to the types of photosynthetic processes, they can be broadly categorized into oxygenic and anoxygenic photosynthesis. The purpose of all the photosynthetic reactions are to generate ATP and reducing equivalents, and also sugars during the dark phase of photosynthesis. Now, coming to the oxygenic and anoxygenic photosynthesis, during like some photosynthetic bacteria, they make use of other electron donors such as hydrogen sulfide in the environment. So when hydrogen sulfide is reduced, the end product is sulfur. And since oxygen is not released during this process, so hence the photosynthesis is known as an oxygenic photosynthesis, since oxygen is not generated in these organisms. While in case of oxygenic photosynthesis, which takes place in case of green plants, algae, and cyanobacteria, They make use of water as a source of electrons, and as a result, oxygen is liberated as a byproduct. Hence, it is known as oxygenic photosynthesis. So here are some of the components of the N-oxygenic photosynthetic mechanism in case of purple sulfur bacteria. The apparatus consists of the light harvesting complex 1 and 2. Then we have the reaction center where the chlorophyll, the reaction center chlorophyll molecule is present, cytochrome BC1 complex, and ATP synthesis. The reaction center is made up of two molecules of chlorophyll A, which is a special pair of chlorophyll A molecule, two molecules of bacteriophyophytin, two molecules of quinones, two molecules of carotenoids, and all these are present or arranged in such a form that the reaction of energy transfer to the reaction center takes place very fast, generating ATP energy. So here is the diagrammatic representation of how the electron transport takes place during an oxygenic photosynthesis in purple sulfur bacteria. So here you can see that the reaction center consists of P870. So the light harvesting complex will transfer energy to the reaction center, as a result of which the P870 gets excited and emits an electron. The electron is first accepted by a bacteriophyophytin molecule, then it is transferred to quinones, quinone A and B, and eventually are transferred to the quinone pool. The quinone pool will accept protons or H plus ions from the cytosol and will get completely reduced to QH to form in the process. So here in anoxygenic photosynthesis, the H plus ions that are required for reduction of quinones are obtained from other compounds such as hydrogen sulfide. So oxygen is not released during the process. From the quinones, the electrons and protons will be transferred to cytochrome BC1 complex through an iron sulfur complex eventually to cytochrome C2. Cytochrome C2 is a diffusible membrane bound cytochrome that will exclude the protons across the membrane during the process, and the electrons are shuttled back to the excited reaction center, and the cycling back of the electron will help the excited reaction center chlorophyll molecule to come back to the ground state. So in this way, there is cyclic transfer of electrons, during which process the H plus ions are excluded, generating a proton motive force, which is utilized by ATP synthetase to generate ATP. So in this method, ATP generation is taking place by cyclic electron transport, and hence the electron transport chain is also known as cyclic electron transport chain. This is another representation of the same cycle, how the electron transport chain takes place. Now, in case of oxygenic photosynthesis, electron transfer will take place to two different reaction centers, photosystem 1 and 2. So the two photosystems are involved here, which are specially separated from each other. Here, ATP generation and reducing equivalent formation are the main purpose of oxygenic photosynthesis. So in case of cyanobacteria, they do not have a specialized structures such as chloroplasts, which are present in case of plants. So in these bacteria, the process will take place on the membranes itself, which will be stacked similar to that of membranes of the chloroplast in the cytosol. So in this case, we have non-cyclic photophosphorylation. So non-cyclic means that whatever electron is being emitted by a reaction center does not comes back to the reaction center. This type of electron transport is known as non-cyclic electron transport, involved in non-cyclic photophosphorylation. The scheme is also known as a Z scheme. So here we have two different photosystems present. The photosystem 2, consisting of P680, it will absorb light energy and emit an electron, which will be first accepted by a pheophytin molecule, and then quinones A and B, eventually reducing the quinone pool. From quinone pool, the quinones will get completely reduced, and then the uh, electrons and protons are transferred to cytochrome Bf1 complex. From here, it is transferred to a plastocyanin, and from plastocyanin, they are transferred to the photosystem 1. The photosystem 1 also simultaneously absorbs a photon of light and gets excited, emitting a photon, emitting an electron. The electron is transferred to chlorophyll AO molecule, then to an iron sulfur protein, then to a ferredoxin, and finally to a flavoprotein molecule. The flavoprotein molecule is involved in generation of reducing equivalents in the form of NADPH. So here, the electron that is emitted by the photosystem 1 is actually used up in generation of reducing equivalent. Alternatively, the electron can also flow from ferredoxin molecule back to cytochrome Bf complex, again cycling back to plastocyanin and to the reaction center P700. So if you look at this, this is like a cyclic path of electrons if only this cycle is active, or only the photosystem 1 is functioning in the microorganism. But if both photosystems are active, then you have a non-cyclic flow of electron, and whatever vacuum or gap is created in the photosystem 1, that is fulfilled by the electron received from plastocyanin, and whatever gap or vacuum is created in the photosystem 2, that is filled up by the electrons that are obtained from breakdown of a water molecule. So as a result, oxygen is released during the process, and hence it is also known as oxygenic photophosphorylation. Lastly, coming to CO2 fixation by Kelvin cycles. So again, this will happen only in case of photoautotropy. If fixed form of sugars are already present, then the Kelvin cycle will not take place in case of organisms. So the requirement for Kelvin cycle includes ATP, NADPH, enzyme ribosco, which stands for ribulose bisphosphate carboxylase, and enzyme phosphoribullokinase. So the ribosco enzyme catalyzes formation of two molecules of 3-phosphoglycerate using one molecule of ribulose, 1,6-bisphosphate, and carbon dioxide that is fixed during this step. Later, 3-phosphoglycerate is phosphorylated to 1,3-bisphosphoglycerate that is using up ATP during this step. 1,3-bisphosphoglycerate is then further reduced to glyceraldehyde 3-phosphate utilizing NADPH. And from here, glyceraldehyde 3-phosphate is converted eventually into glucose or the 6-carbon sugars by the steps similar to that of the reverse steps of glycolysis. So if, overall, you can say that 6 molecules of CO2 are fixed to generate 1 molecule of 6-carbon sugar, during which process 12 molecules of NADPH and 18 ATP are utilized. So that's it about cyclic and non-cyclic and oxygenic and anoxygenic photosynthesis. Kindly like, share, and subscribe if you like the contents of the video, and to get direct access to the videos uploaded. Thank you.